Today, I am taking up the topic search for the genetic material, that is DNA. It all started in the year 1804 when the nucleus was discovered by Robert Brown. Then later on, another scientist known as John Frederick Misher in the year 1871 discovered DNA in the sperm cells of a fish. He called it as nuclein. Then, in the year 1903, chromosomes were discovered as units of inheritance. Then, in 1910, it was discovered that genes are arranged in a linear order on the chromosomes. So they are arranged one after one throughout the length of chromosomes. Then, which one is the genetic material? Chromosomes contain genes. Then, chromosomes contain proteins along with the DNA. Then there was a dispute among scientists which one can be the or could be the genetic material. Proteins are very complex structures and there are millions of types of proteins. Whereas DNA, structurally it is very simple. Very simple. So everybody thought that the genetic material should be very complex. It cannot be a simple substance. So the scientists were inclined to believe that proteins are the genetic material and not DNA. Then, in the year 1828, Frederick Griffith conducted one wonderful experiment to show that DNA is in fact the genetic material, but nobody believed it. So in the search of genetic material, 3 experiments are very important. The first one is transformation experiments conducted by Frederick Griffith in 1928. Then identification of the transforming principle by 3 scientists, Oswald Avery, Colin MacLeod, and McCarty. The third one, the final proof, comes from the experiment of Hershey cheese in the year 1952. So these are very important. So the turning point in the identification of DNA comes from the experiments of Frederick Griffith in the year 1928. He used the bacterium Streptococcus pneumoniae, which causes pneumonia in human beings. In mice, it causes death. So, he selected the bacterium Streptococcus pneumoniae for his experiments. Streptococcus pneumoniae occurs in 2 forms. One form is capsulated, other one is non-capsulated. Here what happens is, the capsulated forms are covered by a gelatinous capsule. They are covered by a gelatinous capsule. So, the colony of these capsulated type appears smooth on the surface. So, they are known as S type. They are also known as S type. The other one, non-capsulated, they don't have any gelatinous coat made of polysaccharides, and their colony appears rough. So these are known as R type. So S type, R type. Then the capsulated ones, as they are protected by the polysaccharide capsule, they are immune from the action of the defense mechanism of the mice. So they survive and cause death. Whereas the non-virulent or non-capsulated ones are non-virulent and they are destroyed in the body of the mice. They can't do any disease. They can't cause any disease. So, capsulated ones are very dangerous. They are lethal, and they kill the mice when injected. But non-capsulated ones, they cause no death. Then, Frederick Griffith actually used type 3S type and type 2R. Type 3S is virulent. Type 2R is not virulent. He conducted a very simple experiment. First, what he did, he injected non-capsulated type into mice. And what is the result? Mice survived, they have lived. It is expected. Then he injected live capsulated type into mice. Then mice died. This result is also expected. Third experiment, he injected heat-killed capsulated type into mice. When you subject the bacteria to heat, they die. So, naturally, mice survived. So, these three experiments are expected. The results of the three experiments are expected. The fourth one is very interesting. Only a true scientist can carry out such experiments. Even such idea doesn't come to our mind. It's very, very extraordinary thought. What he did, he heat-killed capsulated type. That means he killed them. Then he added live non-capsulated type to that. And that mixture was injected into mice. So what he did, he selected heat-killed S type, added to them live R type or non-capsulated type, and both of them are together they are injected into mice. Everyone expected that mice must survive, but on the contrary, the mice died. This result is quite unexpected. Nobody expects a result like this because the killer variety, the capsulated ones are dead because they are heat killed. But this is unexpected. Griffith didn't keep quiet at that level. He cut open the mice, and to his great surprise, he found inside the mice live S-type bacteria. That means R-type have transformed into S-type, or non-capsulated type transformed into capsulated type. How it occurred, he didn't know. He couldn't understand. And he left the experiment at that level. Because of the transformation of R type into S type, this experiment is known as transformation experiment or transformation experiments. It has great significance in identifying DNA or genetic material. Then, in the year 1944, three scientists came forward to analyze what is the transforming substance, how the R type is transformed into S type. So the three scientists were Oswald Avery, Colin MacLeod, and McCarty. What they did, they continued the experiments on Streptococcus pneumoniae. First they have killed the S type, the lethal type, by subjecting them to heat. Then these bacteria are covered by, I have told you, a gelatinous polysaccharide coat. They have removed that coat, gelatinous coat, by using suitable enzymes, and they have extracted the active portion from the bacterium. So once the gelatinous coat is removed, the leftover part is called active portion. The active portion has three parts: proteins, RNA, and DNA. Then they conducted a classical experiment on this material. First, they have subjected the proteins to enzymes proteases like trypsin, chymotrypsin, etc. When you subject proteins to proteases, what happens? Proteins are digested and amino acids are produced. That means proteins become ineffective or they are destroyed. Then the active principle, that means active principle minus proteins, that means still contains DNA and RNA, was injected into mice. And they found that mice died. Then they used RNAs, enzyme RNAs on the active principle, active portion. RNAs digest RNA. Then they injected that into mice and mice died. Here they have added R type also. So in these experiments, they have added the active principle to R type and together they have injected into mice. Mice died again. That means proteins are not genetic material. RNA is also not a genetic material. Then what they did, they treated the active portion with the DNase. DNase is the enzyme that destroys the DNA, that denatures DNA. So when they treated the active portion with the DNase mixed with R type and injected into the mice did not die. Transformation did not take place. When they opened the mice, they found that only R type was found. There was no S type. This experiment has clearly shown that DNA is the only genetic material and not RNA or proteins. This is a classical experiment. But still the scientists didn't believe them. Majority of the scientists still believed that proteins are genetic material. DNA is not the genetic material. They were unconvinced. Then, the third experiment. The clinching proof of DNA as genetic material was carried out in the year 1952 by Alfred, Hershey and Martha Chase. It's a very, very important experiment. The next year, the very next year, the DNA structure was proposed by Watson and Crick. So there is only 1 year gap after this experiment. So they have used the bacterium Escherichia coli or simply E. coli. And they have also used the phase T2 virus. Some virus attacks the bacteria. Such viruses are known as phages or bacteriophages. So in this experiment, Hershey and Chase, they have selected phase T2 virus. Their experiment is very ingenious. In proteins, there is sulfur. In DNA, there is phosphorus. That you have to remember very carefully. So, proteins contain sulfur, DNA contains phosphorus. They have used radioactive sulfur and radioactive phosphorus in their experiment. So, in one container, they have used radioactive sulfur and they used the bacteriophages. In another container, they have used the radioactive phosphorus and they used the bacteriophages T2. So in one container, radioactive sulfur and another container, radioactive phosphorus. So separately they have raised the bacteriophages. Then they have used them in their experiment, which is a classical experiment. So, non-radioactive phosphorus is 31. 32, 35 one is radioactive. Sorry. Radioactive phosphorus is 32P and non-radioactive phosphorus is 31. Similarly, radioactive sulfur is 35S and non-radioactive sulfur is 32S. They have used 32P and 35S in their experiment. Then, when the bacteriophages are used, are grown on radioactive phosphorus 32P, they have incorporated this radioactive phosphorus into their DNA. Then, when the bacteriophages are grown on the container, containing radioactive sulfur 32S, this is incorporated into their protein. So this is very important. So radioactive sulfur is incorporated into the protein. Radioactive phosphorus is incorporated into the DNA. Now, let us see what happens. These are the viruses or phages grown on radioactive 35S. A virus has structure like this. It has a capsule called capsid made of protein. So this is called capsid or protein capsule. So 35S is incorporated into this capsid. Inside there is DNA. Because these phages are grown on radioactive sulfur, that is 35S, that sulfur is incorporated into the protein and they have a radioactive capsid and a non-radioactive DNA. Then they infected bacteria with these phages. Then what happens? The phages get attached to the bacteria and release their DNA into the bacterial body. Once this process is over, these were subjected to a blender, or they were put in a blender which blends. In a blender, the bacteriophages which were empty were separated from the bacteria. Here they are attached, but when they are subjected to a blender, the empty bacteriophages or ghost cells are separated from the bacteria. Then the fluid is subject to centrifugation. In the centrifugation, what happens? Substances settle according to their density. So when they subject to the centrifuge, they got two fractions, fraction 1 and fraction 2. In the fraction 1, we come across these ghost cells, and the fraction 2, we come across the bacteria. Then the bacteria are separated and allowed to multiply. Then what happens? When the bacteria try to multiply, the DNA of the phage multiplies first. And after some time, the phages, the progeny of the phages, break down the body of the bacterium and they come out. They tested the bacteriophages for radioactivity, but to the surprise of Hershey and Chase, there was no bacterial activity in this progeny, because the radioactive capsule was left out. Only non-radioactive DNA has entered their bodies. So the progeny derived from this non-radioactive DNA contained only non-radioactive DNA. Their protein code was non-radioactive because the sulfur did not enter the body of bacteria. Then, in the next experiment, what they did? They have used the viruses grown on radioactive phosphorus. Similarly, they infected the bacteria with the viruses grown on 32P, or radioactive phosphorus. You know that phosphorus is incorporated into the DNA. Here, this is the phase attached to the bacteria, and it releases its DNA into the bacterial body. This DNA contains radioactivity, but whereas the capsid of the virus does not contain any radioactivity. Then, these were subjected to a blender or blending, and the ghost cells are separated. Again, here also you get two fractions, fraction 1 and fraction 2. In fraction 1, we come across these ghost cells. These ghost cells are non-radioactive. Fraction 2 contains the bacteria. These bacteria are allowed to grow, and as they grow, the DNA of the phase multiplies, and a number of daughter viruses are formed, and they come out. Then they come out by destroying the cell wall of the bacteria. When this progeny was tested, they found to exhibit radioactivity. It means DNA of the parent virus, which is radioactive, is inherited in the DNA of the progeny of that bacterium, of that virus. So in this way, this experiment clearly shows that DNA is the genetic material and not proteins. So this experiment of Hershey and Chase conclusively proves that the genetic material is DNA and not RNA, not the proteins. So with that, they have put an end to this argument. That is about discovery of DNA as the genetic material. That is about this topic. In the next topic, I will deal with the structure of DNA. So before I conclude my lesson, just I have one message to the students. See, I have written 3 books that are helpful in NEET examination 2024. One is NEET Zoology 2024, NEET Botany 2024, NEET 2024 Biology Question Bank, which contains more than 5,000 MCQs. Out of the 5,000, I have framed more than 2,500 questions for your practice. These books are available only in the form of Kindle edition in Amazon. Each book costs about 500 rupees. They will help you a lot because these books deal with the subject in a most systematic way for the benefit of the students. Thank you.